is dissection of a bird. So I have in front of me here a chick which is getting to about 2 months old. So we are first of all going to observe the external parts, just like we did. You realize that we have the beak here, which is pointed, sharp pointed, which is used in picking food from the ground. We have the nares, or the external nostrils, that are used for breathing. Then we have the comb up this way, this is the comb. And down here we have the wattle. The wattle here is still poorly developed. Down this way. This is the wattle which is developing. And then we have, this is the eardrum, this is the external ear here, which is covered by feathers, covered by some feathers. And then you have the eye. So we notice that the eye has a very, very movable eyelid. And there's also another tissue inside that cleans the eye, which we call the nictitating membrane. It's not seen because it is closing it. And then we will take note that the comb here functions as an outward expression of sexual maturity in birds. But this one is still a young chick and it is not well pronounced. So we see that, we go to the legs, we see that the leg has 4 digits and the digits are divided. That one is facing behind and the other ones are facing ahead. So the use of these digits facing behind and in front is that it makes the bird to be able to hold the branch on which it is landing. So you can see how it has held my finger. So if it's landing on the branch, it will hold it like this, and you are turning it even upside down, it will still hold it like that so that it doesn't fall off. So this kind of arrangement of digits is what we call the 3+1 arrangement. Because you have the leg that comes like that, you have the other 3 this way, the other 3 up this way, and then you have one behind this way. So it ends with a cloud like that. So this is what we call the 3+1 arrangement. So you have 3 digits this way and you have 1 this way. So this is called a 3+1 arrangement that we have here. You have some birds that have a 2+2 arrangement. Looking at this chick, you realize that this is the tail. And on top of the tail region, you have a gland here. There's a gland up this way. This is a gland here, which is called a green gland. So this gland produces a fluid that is used to clean the feathers and also to make the feather to stick together so that water does not penetrate and reach the body of the chick. So we also see that the legs have scales. The legs are scaly down here. These are scales. And there is a small thing that is growing here. This thing is what is called a spur. It is called a spur. It's growing somewhere here. It's called a spur. So it also functions as a sign of sexual maturity in birds. So we have the wing. If we open the wing, we see that the wing of the bird has special feathers. So this is the wing, and the wing has the special feathers. So these feathers of the wing are what we call the flight feathers, what we call the quill feathers. So they are found on the wing and they're also found on the tail, and they are used for flight. You realize that when the bird is flying and it is balancing, it uses the tail to balance off. If I'm bringing down this one, you see it is turning the tail, so it uses the tail to balance, and these wings are to sustain it in the air during flight. So we also have some other feathers which are called the contour feathers. And these are what we call the contour feathers, these ones that are found on the back here. Contour feathers are found on the back, and they are also found on the neck here, especially in mature birds. They are found here on the neck. They are the contour feathers. So they help for insulation. Then we also have some other feathers which are called the down feathers. So the down feathers are found on the neck region, these ones here. These are what we call the down feathers. You see that they are smaller than the contour feathers, and they are far smaller than the flight feathers. These ones are the flight feathers. This one is the flight feathers or the quill feathers. Then another kind of feather that we have is what we call the covert feathers. And the covert feathers are found under the contour feathers, in order that the wings of the chicks should not allow air to pass. So you see that in this wing, you have these feathers which are found there. So these ones are what we call the covert feathers. These ones here. These are the covert feathers which are found in between the flight feathers of the wing. Then we also have the phyloplumes. The fourth kind of wing of feather is the phyloplumes, which are very visible only in all fowls. And you see that the phyloplumes have no precise biological function. But the other kinds of feathers they function for insulation and protection against mechanical injury, and the flight feathers, they function for flight. So you can be asked to take the chick, dissect it, and to place the wing and draw it. So if you to describe only the wing, it means that you place it as such, pin it, and then you draw just what you have had. So when you look at it, you see the various muscles that are found on the wing that makes the bird to be able to fly. So you also see that there is another end this way, which we call the bastard finger. This end this way. There's an ending this way, this part here. This is what we call the bastard finger. So this bastard finger makes the bird to be classified as having a primitive pentadactyl limb because of this. So this is synonymous to the thumb in the higher animals. So we are going to go straight into the dissection of this chick, and the first thing is we are going to kill it. So I'm going to kill this chick using my hand. So I'll kill the chick by beating it on the occiput. So I'll beat it here, and you'll see how a chick dies. So when you beat on the occiput, you first of all destroy the connection in the nervous system, and you beat again, the thing will just begin to die. You see that it is almost dying. You beat again just with the finger. You beat like that. So the thing is almost dying. So this region where I've beat it and it is dying is what we call the occipital region. Just like in humans, it is not advisable to beat somebody, especially with a hard object, at the occiput. Because at the occiput, it is where the hindbrain joins with the spinal cord, and also the main blood vessels that carry blood up into the head link at the occipital region. So when you beat somebody at the occiput, you dislocate the nervous tissues that enter into the brain, and you also block the blood that is passing into the brain, and also you can equally dislocate the joints, because the skull bones join with the bones of the spinal cord at the occiput. So when these are dislocated, somebody can go into a state of a coma that's becoming numb, not sensitive, and eventually that person can die. So it is advisable never to beat somebody, especially with a hard object, at the occipital region. Because this is what we have done, and we have seen what is happening to this bird. So it can equally happen to humans. So we are going to go straight into the dissection of the bird, and we first of all pin it onto the board. This is how we pin the chicken. Remember, in the guinea pig, we only pin the limbs. So for the chick, we start by pinning the beak, and then we pin the other side. But we cut the feathers of the wings so that it should be quite clean. But here, we do not wet it. So what we do is that we have to shave. But shaving does not take a long time. But if you were dissecting a mature bird, you can simply remove the feathers. But since most often, what we dissect are very young birds, very young chicks, then we just have to cut it with the scissors. So we just have to clean it with the scissors by shaving. So you must be able to shave these feathers. These are what we call the contour feathers, which function for insulation. So I'm shaving it. I come down here, I shave this one. Shave it. So it has to be clean. It has to be shaved. Because if you don't shave it, your dissection will not be clean. So it tells us that we should always have a small waste bin by our side when we are going in for our exams, because you don't know what will be provided. You don't know what will be done. And the incision which is made in birds is made right down here at the abdominal region. This is the abdominal region. So make the first incision down like that. So this is the first incision which we make here. Make it down to cut off the outer skin. And after we make the incision, we can increase it, holding it with the hand, can increase the incision as such. So when we cut it, we see that there's still another inner tissue. So we'll still make the second incision. So we'll make the second incision to cut off the abdominal wall. And when we cut it now, we can then be able to see the contents of the abdomen inside. So when we see these contents, we will have to increase this incision coming down this way. This is the cloaca opening. So increase coming towards the cloaca, but we do not destroy the cloaca. We just simply open the small abdomen. So we know that the abdomen here is quite small. So cut it also this way, a little bit this way. Use our prop to trace the part that we are going to cut. We are not going to cut up like this as we did in guinea pig, but we are going to cut now on the sides. So we're going to first of all to cut on the left hand side like that. You can first of all cut this tissue so that it makes you to be able to see what you are doing. So when you cut this tissue up, you clean it and you'll be able to be seeing what you are doing. So you can cut it right up to this level, but you should be careful not to cut the esophagus or to cut the trachea. So we clean up this tissue like that, clean this tissue so that we can be able to see what is happening. So when we clean the tissues, we end this way. We end here. Because if we don't end here, we'll destroy something up that way. So clean and end here. So when we clean it, you see that this is the xiphoid cartilage, and these are the pectoral muscles, the muscles of the chest region. So the dissection goes as such. We add some water. It's already getting dry. We add some water, and then dissection goes as such. We start cutting from the right hand side and cut this way. We cut like this. I'm cutting the ribs. There's a place where the ribs bend down this way, and if you are cutting only at that bend, you cut it with ease, and the ribs will only cut as if they are not strong. So we also cut these pectoral muscles this way. Then we cut this bone that passes this way. There's a strong bone here, which we call the coracoid. So the bone is up this way, so I cut it, and I'll cut and open up this way. So I have to cut this strong bone, which we call the coracoid. So I'll cut it this way. Up now onto the left hand side. Come now onto the left hand side, and I also cut it the same way as I was cutting onto the right hand side. So you see that I am at this juncture cutting the right hand side of this bird, and I'm going to cut it, and it will link onto this region right below the neck region. So I'll cut it up, cut this. I cut the coracoid, this one. I cut it. I cut this tissue here. So after cutting it, I will remove this tissue. I will remove this tissue. I'll remove this flesh that I've cut and throw it. Then, when I cut like this, you see that these automatically are the abdominal contents of the bird in situ. We can see the heart here that it is still beating. You can see the heart still beating. See the heart. So this one is the right atrium, and this is the right ventricle. This is the left ventricle, and this is the left atrium. So you can see blood vessels that are coming to the heart very distinctly. You can see the septum. You can see the septum which is coming, and we also have to cut up this way. We also have to cut up this way so that we will be able to see the trachea as it comes down. We have to see the trachea this way. So we'll cut up the tissue this way and we'll throw it. So when we cut it, this is the bone which is called the coracoid. This is the trachea. This one is what is called a crop, where food is stored. So these are the contents in situ. This is the heart, this is the liver, and then this is the gizzard. This part here like this is the duodenum, which is very visible. You can see the pancreas there, very visible, and these are the intestines. So straight away, when you cut it, if you were to display the alimentary canal, what you simply have to do is, remember to always weight our dissection. What we have to do is, we have to take out the intestines as they are, and then we start opening them, preferably from behind. So we start opening them from behind as such. So once we start opening them, you see that I'm holding them like this. I'm opening and coming up. I'm opening and coming up. So I place it onto this side. These are the mesenteries. Place this way, mesenteries, and then cut this mesenteries. But when I'm reaching up here, I see that the duodenum is very distinct, and you see that the pancreas there is very distinct with the pancreas inside. Very distinct. This is the gizzard. I place it this way and I put it like this. So if I put it like that, I'll open the liver upwards like this, so that I will be able to see. This is this. I put it like this, so that I will be able to see the spleen right here, and this is the liver, and this is the heart. So if I display it like this, you see that this is the display of the alimentary canal. This is the crop, put it this way, and this here is the gullet. This is the gullet here, or the esophagus, where food comes down, and it is temporarily stored here in the crop. When it passes from the crop, it comes down inside here into what we call the blue stomach, or what we call the blue ventriculus. Inside here, we are going to see it a little later. It's over. And then it passes then into the gizzard. So the gizzard contains some sand particles and hard particles which help to break the sick coats of the cotyledons, so that enzymes can then go into the particles or the grains that the fowl eats and then digest. In situ, we have dissected and displayed all the systems which are found in the guinea pig, and we are also going to isolate the thoracic cavity, where we have seen and isolated the main part. We are going to the digestive system, and we are going to the genital system. So candidates should take note that the question can be asked which requires all of these systems to be drawn or to be displayed at once, and it can also be asked for two or one of these systems. But you should take note that if you are asked to give those one, to display one of the systems, the other ones are not necessary. So you take the other ones off, so that the one that you were asked, you isolate it and let it stand out very distinct. And you also make sure that you draw a diagram to look very much like what you have done. Don't draw what you think. Don't dissect a female organism and draw a male organism because you have dissected a male organism during your class lesson. So if you dissect your organism and you are drawing it, it is good to write the sex there. For example, you write that the thoracic cavity of a male guinea pig, or the genital system of a male guinea pig. That is obvious. So if you write a male guinea pig and the examiner says that you have dissected with a female guinea pig, then you are wrong. It means that you don't know what you are doing, you lose much in life. So you should always be able to write on the sex of the organism in line with. We now see that in dissecting a bird, when we remove the abdominal contents, the intestines, and put this way, you see that this part is empty. Then up here, I'll add water to make it wet. Up here, we see the heart that is there, and then we see the gizzard this way. But first, I will like to keep the abdominal content first intact, and then I will display the thoracic cavity. But there is one thing which is very outstanding with the bird. We have seen that in the guinea pig we had the diaphragm, but the diaphragm is conspicuously absent in birds. So this shows that the birds are still less advanced evolutionary to mammals. That's why they are developed the diaphragm to separate the abdominal cavity from the thoracic cavity. So we have seen that it is just continuous like that. But in the mammal, everything was not continuous. It was separated by a muscular diaphragm. So I can still cut this one down a little bit so I will see it more clearly. I cut like that. So see that it is standing out clearly. So from here up to this level, we put up. We are going to, first of all, we have already explained the contents of the thoracic cavity, and we are going now to display the alimentary canal. So I will display it, and the thoracic cavity will remain intact, and you will see what we can do. So you realize that I'll put this duodenum this way. So we see that this one here is the gallbladder. This one that is green in color, this is the gallbladder, where bile is stored prior to elimination to the duodenum for digestion. And then here is the liver. This is the heart that we are seeing. We'll turn the liver the other way. We'll see a spleen, and the spleen is quite small. So we put the duodenum this way, and then this part now, which is the ileum, we can now display it onto our right hand side as such. Display it, making sure that it is in a very nice looking manner. And down here we see some two branches. So we'll cut these branches. We are going to separate them like that from the alimentary canal. They too are part of the alimentary canal. So we are going to cut it, and we'll put it this way. And then you see that these two branches, these are what we call the digestive diverticuli. These two are what we call the digestive diverticuli. So it functions in the way that when the food is digested along the alimentary canal and it comes here, and there's still some nutrients and especially water, the food comes up into this digestive diverticuli, and when it stays there for some time, much of the water is absorbed. That is why you realize that birds send out feces in the hard form, and their feces are hardly watery, except the birds are sick. It's because the water, much of the water and the rest of the nutritive part of the food is absorbed at the digestive diverticuli. So you see that it joins down here, and this is now where feces is stored. This is the rectum, feces is stored here, and eventually it is eliminated here. So we now see that there is no opening out for the elimination of urine, and we know that the birds send out their urine or their nitrogenous waste in the form of nitric acid, which is hard and whitish. What whitish you find mixed with the feces of birds, that's the nitrogenous waste that they send out along with feces. So if you display it as such, you now realize that this one will be the display of the alimentary canal of a bird. You make sure that this crop stands out distinct. This crop stands out distinct, and you can pin it. You can always pin it onto your board this way. You can always pin it onto the board like that, so that you have the esophagus coming down, entering into the crop where the particles of food and grains are stored, and then it comes down here. Right inside here you have the proventriculus. This is the proventriculus here. This is the proventriculus inside under the heart, and then you have the heart here. You have the liver loops. This is the liver. Then you have the gallbladder here. Then you have the duodenum with the pancreas. Then you have the small intestine that comes up this way, and you will note the conspicuous absence of the large appendix. This is a small appendix which is found here. This is the appendix, and the appendix is small, just like in humans, indicating that the birds do not contain cellulose digesting bacteria. So this is how an appendix of a human being may look like. So it is also reduced like this compared to that of a guinea pig. So when you display as such, you put up a diagram of the alimentary canal, which looks as such. Diagram of the alimentary canal, okay, that looks as such. So on this diagram, you see that is a bird that has been dissected, and the alimentary canal has been displayed. So you see that this is the head region up this way. So you have the head region up this way. This is the head region that we have here. We have opened out this because we have allowed this, but on this diagram, the other parts are not allowed. Then you see the crop. This is the crop that's indicated here. You see the duodenum with the pancreas. Then you see the liver loops, and then you see the gallbladder, then you see the small intestines, you see the mesenteries, and then you see the digestive diverticulum, which is here, and you find the colon here. Then you have the cloaca and the anus. So all of these parts on that board on the board, you can see them in this, our dissection. These are all the parts that are there. So you draw your diagram to look like what you have done. You label, this is the cloaca here, this is the colon, and all of these are there. So you draw it and keep it clean. The examiner will always give you your marks. So another aspect is to display the urinogenital system. To display only the urinogenital system. So in that case now, I am going to remove the alimentary canal. So I will cut off the alimentary canal. I will cut it off. The alimentary canal, I'll cut off carefully, not to destroy the other parts which are still necessary. Remove it. I cut off from this way. Cut it off like that. So I cut it. So I'll cut it off. I'll cut from the gizzard. Now pull it out. Okay. So when I pull it out now, we can be able to see that true stomach. This is the true stomach which was found under the heart there, which we did not see. This is the true stomach. It is very small. This is what is called the true ventriculus. This is where digestion starts here. So if we open this true ventriculus, we'll be able to see that the cells there are able to secrete digestive enzyme. They're able to secrete gastric juices. So I'll open it like this. I'll wash it, water. I wash it, and then you see that if we press it, these walls, you see a fluid coming out. I will press it. I press it like this. You see a fluid that is coming out, which is like milk, milky, from this indentation, these things that look like inflammations. So these are the gastric glands that secrete the gastric juices. So you can see the fluid. This is the fluid that has come up like that and gathered. I can put it here. That has come up and gathered. It's whitish like my glove, so we may not be able to be seeing it. So it comes up from these indentations here. So these are the glands in the stomach walls that secrete the gastric juices containing the various enzymes as we know. So we'll keep this one now into our waste bin. Then take off the blood. Blood is not supposed to be there. And then we also remove the heart. We remove now the liver. We don't need the liver. The liver is part of the digestive system. So this is the spleen, the small spleen is there. Remember to always clean it. So as we clean it, we see that what we are left with here now, what we are left with here, we have the heart here. So this is the circulatory system. For the circulatory system, what we have to present is the heart and then blood vessels. So I have to present the heart with blood vessels. These are blood vessels like this. And then we can also see the lungs. These are the lungs. We call them the pocket lungs, where the lungs are found inside the muscle, the bones of the heart. These are the lungs on the sides, like this. These are the pocket lungs on the side. So this is the circulatory system. So from this diagram, we'll see clearly that this is a cock. This was a young cock, because these are the two testes. These are the testes here, like that. So if it were a female, we'll find ovaries here with a lot of primary follicles. That is very tiny eggs hanging here, many of them, like that. So these are the testes. This is where we see that it was a cock when it is still in a young stage, like this. So with this, we can put up a diagram of the circulatory system. This is the circulatory system. The diagram will look as such. The circulatory system, you see that we have already opened up, and we have the heart, which is down here. This is the heart that we have here with the blood vessels. So we can draw it to look something like this. So this is a diagram that was drawn. See that this is the heart standing out, and you see the blood vessels. So when we are talking about the circulatory system, our stress is on the heart and the blood vessels. You should be able to identify and locate at least 1 or 2 blood vessels to actually show that you know what you are doing. Then we'll go straight now to displaying the respiratory system. So in displaying the respiratory system, I'll cut off the heart because the heart is not part of the respiratory system. So I'll cut it off, and then what will be left there or the thoracic cavity is just the respiratory system. I don't have to go any far, just cut off the heart, and I will be able to see the trachea. So you see that this is the trachea that has come here with rings of cartilage. This is the trachea here, and you see it branching. This is it branch here. This is it branching here towards the left lung, and this one branching here towards the right lung. So these are the lungs. These are all about the breathing system or the circulatory system in the domestic bird. So we can put up the diagram. You see the pocket lungs that have come down in many groups. So we can put up the diagram. It looks as such. So this is the diagram. You can see that this is the diagrammatic representation of the position of the breathing structures and air sacs in the bird. So these are just the structures. So I clean it and let it lie there, and it looks nice. So when it is clean and there's no blood, then your dissection is described as good, and this is just all. So the last thing that we are now to look at on the bird is to look at the urinogenital system. So to look at the urinogenital system, we do not even need to remove the lungs, but we can equally remove the lungs, which is quite simple to remove. You just simply tear them up from inside these pockets. You can do it with your hand or with the help of a scissors. You tear them up like that. You remove the lungs. Remove the lungs, and equally we can remove this trachea. You don't need it anymore. Remove the trachea, and then you realize that what you are left with directly here, what you are left with is the urinogenital system of a bird. It is not as complicated as in mammals. So this is just simply the urinogenital system of a bird. So you see that these are the kidneys that come down like that. These are the kidneys. The kidneys that come down, these are the 2 kidneys. And these are the testes. So the testes have the sperm ducts which come this way. They are quite small. These are this one, and this is the other one this way. They come down here, and they come and join here at the cloaca. They come and join right down here. You can see the junction this way. This is it, that they come and join down here. They come and join. So the ureter also leaves the kidneys and come and join here. So this is just all about the urinary system of a male bird. So when you do like this and clean your dissection, you simply have to draw a diagram that looks like what we have here on the board. So you draw a diagram. That here on the board we have this is the diagram. Remember to put the outline. You have, these are the testes which are found up here, these small ones here, down them here. And then you have the kidneys. You see the kidneys down here. So you have the vas difference, or what we call the sperm duct, and then you have the ureter which carries the urine from the kidney coming down, and you have the urinogenital opening, or what we call the uridium, which is down here, and the link that it has with the cloaca right down there. So this is just all about the urinogenital system of a male chicken. So if you do like this and draw a diagram, that is just all. You don't have to waste time with dissection of the bird. So this is just all that you have to do. The bird is quite simple, straight to the point, and it takes very less time to dissect and display any system of any choice.